Hi, my name is Blaise. I'm a research technician and part-time PhD student here in the Centre for Stem Cells and Regenerative Medicine at KCL. I moved to London from St Lucia and then completed my undergraduate degree in biomedical sciences at Royal Holloway University. I then graduated and joined the lab here as a research technician, where I'm responsible for everything from glassware washing and autoclaving to media prep. I picked up loads of other duties though, so I also manage all of the histology in the lab. I cut fixed and frozen tissue sections, and I train all the users on our histology equipment. I didn't have much lab experience before I came here, but I think my job as a technician has really helped to serve as a great entry point for my PhD. So early this year I started working on a PhD in Fiona's lab. I'm looking at recreating epidermal niche conditions in vitro, and currently what I'm doing is I'm looking at the effects that different cells in the epidermis have on neighbouring cell fate. I'm chemically engineering the cell membrane with a small molecule called biotin, and then using the strong bond formed between biotin and avidin to artificially and rapidly create cell-cell contact. I get asked quite a lot if it's difficult to study and work at the same time, and at the beginning I did find it quite challenging, but I think as with anything it's a learning process, and now 8 months into my PhD I do find it quite easy to ensure that the basic fundamental tools are in place for everyone in the lab to get their jobs done, but also ensure that my experiments get done on time. In terms of my future scientific career, I think that I'm still quite green and I have a lot of ideas and I constantly hop between what I want to do. But I do really enjoy having a role in both the technical and research side of the lab. So I'm thinking that a career that encompasses both lab management but also allows me to have a role in research would be pretty ideal. Thanks.
I am Atefeh Mobasseri, a biomaterial scientist with biomedical and chemical engineering background. The main key of my research as a postdoc researcher at WattLab at Center of Stem Cells and Regenerative Medicine is to explore cell biomaterial interaction. I have some experience in developing biomaterial and modifying its property to enhance cellular behavior, and also translating this technique from laboratory into clinical trial. I'm hoping to use this expertise to look at the effect of the biomaterial surface on epidermal human stem cells. And we are aiming to look at the effect of the biomaterial surface on stem cells' properties, position, and differentiation. This research project aims to address a significant but basic biological question, which is significant into healthy aging.
Hi, my name is Kif Liakath-Ali. I recently completed my PhD with Professor Fiona Watt. In my PhD project, I identified novel genes involved in skin function using a high throughput, large-scale phenotype screening using hundreds of mutant mice. In this project, I used a technique called epidermal whole mount imaging using confocal microscope. I identified 50 new genes whose integrity is essential for normal skin function. I also discovered these genes are involved in core signaling pathways and are, more importantly, they are associated with human diseases with skin phenotype. This work in general allowed me to also discover genes involved in differentiation and pigment function. And by this project, I have developed a rich hypothesis generating resource that would be helpful for the wider scientific community. Right now, I'm continuing my position as a postdoctoral fellow in the same lab, where I'm involved in 2 projects that focuses on different aspects of skin biology. The one is on gene mutant where a gene is deleted which is involved in synthesis of bioactive lipid compounds in the skin. My investigations led to identify a systemic metabolic phenotype caused by a differentiation phenotype in the skin. This work is now ongoing, and I will be able to complete soon. The second project I'm involved is completely different aspect in skin biology, where I focus on a post-transcriptional pathway gene where which is essential for the skin function. So currently I'm also working on this project, and which is I'm excited with the preliminary results. Now My educational background is zoology and genetics, and my experimental expertise is genome manipulation using various molecular biology tools. I have skills in imaging using confocal microscopy to image the intricate structure of mammalian skin. By combining this experience and skills, I would like to utilize these skills in future and my current project to tackle some of the fundamental questions in stem cell biology, where I specifically would like to address the complex mechanistic aspects of stem cell function in general. Thank you.
Research associate in Hiratsuka laboratory. My research career started when I was a medical student in Osaka University, Japan. There I had basic research trainings, and it was very exciting and creative, so I decided to be a basic researcher, not a clinical practitioner. In my PhD, I learned microscopic skills to visualize growth signal in individual cells. What I found by the technique is that the growth signal in skin cells is not silent in steady state. Instead, there were many local growth signal activations that propagated locally to the surrounding about 200 of cells. We named this spread. Interestingly, spreads were only seen in living tissue. This means some key factors are lacking in cultured cells. That's why I moved to this Hiratsuka laboratory. The lab is specialized in skin stem cell culture, and recently, in collaboration with bioengineering, they use the artificial substrate that mimic actual skin structure. With this approach, I aim to understand skin biology from a new perspective. I expect skin structure is maintained by cell-to-cell propagation of growth signal, and the disturbance of the signal can lead to pathogenesis such as cancer.
Hi, my name is Angela. I'm a computational biologist at Professor Fiona Watt's lab. I have a highly multidisciplinary background. I studied biomedical engineering back in Portugal, and it was a really cool degree because it gave me the opportunity to learn from basic maths to programming and medicine. And it also allowed me to acquire a very important skill, that is to speak with both engineers and clinicians alike. And they don't quite speak the same language, so I think it's something very good to have. And at the end of my degree, I was still fascinated by intersecting physical sciences with life sciences. So I decided to focus by doing a PhD in systems biology. During my PhD, I combined experimental work with mathematical modeling and big data set analysis to tackle the problem of drug resistance in cancer. I've always been fascinated by cell-to-cell interaction in multicellular organisms, and especially by how the cells stick to their developmental commitment. So to further study cell fate, I contacted Fiona to join her lab. Currently, I'm collaborating in a couple of projects here that address stem cell growth and differentiation in the skin, and I aim to be able to generate a comprehensive model of stem cell fate commitment in the epidermis. My long-term goal is to become a professor, but I felt I still needed to acquire more experimental cell biology knowledge and also in vivo animal work, which I have never done before. So with all these complementary skills that I'm acquiring during my postdoc, I hope that by the end of my time here in the center, I will be in a unique position for a leading academic career.